Hello, everybody, and it would be fantastic if afterwards, when we're having the discussion, it'd be great to see your faces if the bandwidth is okay. But it's a great pleasure to be able to participate in this Heteroresistance infectious diseases meeting, which I have once talked on, I don't know, some 7 or 8 years ago in person on antifungal stewardship. But of course, things have moved on, and I'm going to try to, over the next 45 minutes, give you a gallop through this field and also some of the work that I have been doing and our group has been doing in this area. So before I start, I just want to make a couple of declarations. One is that some of you already know me, and I have a great fondness for Cape Town and South Africa, which I really feel after Croatia and the UK is my third home, having spent 2 years there at the now sadly no longer existing GF Jooste Hospital, doing a study in cryptococcal meningitis and HIV, doing lots of lumbar punctures. And here's my lovely research nurse, Fungi, who was a great companion with Graham and Kevin Reeby and Mark and Gary, etc. So very happy to be chatting to this audience, building on the same theme. So, but I just want to say another thing, which is I don't want to, I'm very wary of coming in as an outsider, if you like, talking from London, UK, as some so-called expert on this subject, when indeed we have on this call, and fortunately for you in South Africa, a wonderful expert in this field running your mycology reference laboratory, and actually unique in the world, I would say, in terms of his engagement both in clinical guidelines and implementation and also research. So Nilesh is just such an asset, and we don't have such a person in the UK. We do have a reference lab. Okay. Hello, can you hear me?
Yeah, we can, sorry. Please go ahead.
So yes, I just want to say that Nilesh is just an absolutely fantastic asset, and you'll see that some of he did send me some South African specific documents, which I will refer to just frame our discussions, because there's nothing worse than giving a talk saying, you know, this is how we do it, or you know, this is our context, but actually not thinking about what might be relevant to the people you're talking to. So moving on to fungi, of course, you all know that I'm very interested in mycology, and obviously there are a broad range of fungi that are human fungal pathogens, both the ascomycetes and the basidiomycetes, including my favorite fungus, Cryptococcus. And what is the characteristic, of course, of these pathogens is that they are widely prevalent in the environment around us, in soil, in decaying vegetation, tree bark, and we are all exposed to them through inhalation. And generally, if we're immunocompetent, these fungi remain contained in the lungs and generally don't cause disease or just asymptomatic infection. We also, of course, have Candida species colonizing our guts. And again, it usually requires some sort of breach of the gut mucosa, also in the setting, of course, of antibiotic use, leading to overgrowth for these species to cause invasive infection. Now, we do have a number of different antifungal drug classes, although, of course, there's a paucity of antifungals compared to antibiotics. And indeed, the development of antifungals first started with the drug Nystatin, which we now mainly use for topical treatment, developed at the New York State Department in the mid-1950s, hence its actual name, Nystatin. This followed amphotericin B, which of course we all widely use and love, a very broad spectrum antifungal. Then later in the 70s, 5-FC, but of course it was the advent of the azole class as an oral class that really transformed our management of invasive fungal infections. And then more recently, of course, some more safe formulations of amphotericin, and then also broader spectrum mold active azoles, and then the echinocandins. So, and of course, there are a number of other, including fortunately some new drug classes in the pipeline going through human trials. Over the same period, we've seen a widening spectrum of immunocompromise that's been, of course, engendered mostly by interventions that we humans have developed, including, of course, organ transplantations, also pioneered in Cape Town at Grooteskuur for cardiac transplantations, also chemotherapy for hematological malignancies, intensive care, increasingly older, more frail people surviving for longer with more tubes and lines and longer stays on ICU, of course, the HIV epidemic, and then also more recently, the development of targeted biologic treatments for autoimmune and inflammatory diseases, also rendering hosts susceptible to invasive fungal infections. So in these largely immunosuppressed hosts, for whatever reason, we use antifungals in different patterns. We either use them for prophylaxis, usually in these at-risk hosts, either on ICU or hemato-oncology patients, empiric in patients with risk factors, preemptive therapy when we've got a biomarker, an early diagnostic test available, and then targeted treatment when we've got positive culture or histology. And I've outlined, the main classes that we all tend to use in these settings. And you can see here that azoles feature throughout. There's a great use of this oral class, and the others, of course, the polyenes and the echinocandins, we can only at the moment give IV. We also use azoles for prolonged maintenance treatment, such as in patients with cryptococcal meningitis and HIV, or with patients with chronic pulmonary aspergillosis until either clinical resolution or reversal of the immunocompromise. So we use them for long periods of time. And we also use azoles in crop spraying as fungicides in the environment. So this dual use of azoles, both in the environment and in the clinic, has meant that over the course of time, and this is a slide courtesy of Matt Fisher at Imperial, there's been increasing reports of resistance to all antifungals, but particularly azoles in both human, animal, and plant species, which is a real issue in terms of the evolving problem of antifungal resistance. And indeed, in parallel with the deployment of any new agent, as we all know, as infection specialists, almost invariably, resistance will be reported to these agents, which are largely used as monotherapy. So we see here that increasingly in the context of the HIV/AIDS epidemic, there was evolving fluconazole resistance because of prolonged treatment of mucosal candidiasis, then subsequently itraconazole resistance in aspergillus reported in patients on long courses of therapy for pulmonary aspergillosis. More recently, FKS mutations in the target gene in resistant glabrata, then also environmental azole resistance, leading to clinical azole resistance, and then of course Candida auris identification, which we'll talk about later. So of course resistance is not the only reason why treatment fails in invasive fungal infection, or indeed any infection. And there are other factors which, such as host immunosuppression, drug pharmacokinetics, patient adherence, and also, as we all know, source control, particularly for Candida, and removal of plastic or any kind of biofilm material. But antifungal resistance, either intrinsic or acquired, is certainly one of the reasons. But not only that, but all of these other factors really do feed into development of certainly acquired drug resistance, which is why we do need to consider them all when we prescribe antifungals. And rather than, do the conventional talk on the different mechanisms of resistance, etc., which I'm sure this audience already knows the basics, I thought I would illustrate antifungal resistance through two cases, clinical cases. The first one is, of course, the disease closest to my heart that I spent 2 years studying in Cape Town between 2004 and 6. This patient is actually from Tanzania, where my former PhD student Neil Stone did his research in Dar es Salaam. And this was a typical patient presenting with a subacute onset of headache and confusion and newly diagnosed HIV with a very low CD4 count, concurrent other fungal infection, candidiasis, which may, of course, in some cases mean that patients have been exposed to azoles before. Anyway, this patient had never been treated with azoles and had a lumbar puncture. CSF was found to be India ink positive. And as we know, as in much of sub-Saharan Africa, and indeed in Cape Town, when I first arrived in 2004, there was no amphotericin B available, not even deoxycholate. So the standard treatment was given of 2 weeks of fluconazole, 1,200 milligrams, then 800 milligrams consolidation, followed by 200 milligrams long-term treatment. And we know from our work with Graham and colleagues at Yorster Hospital, this is a study that I did on the left, that amphotericin B is rapidly fungicidal, whereas fluconazole at the doses we used to use back then is essentially static. So this is looking at the fungal burden, doing serial lumbar punctures in patients over time and plating out onto fungal media and counting the colonies. And you can also see from a later study that even this higher dose of 1,200 milligrams a day that Nikki Longley did in Uganda, and she also subsequently, of course, many of you will also know her from Cape Town, showed that it's only poorly cidal. So it doesn't sterilize the CSF during that induction period when you give 2 weeks. So there is something known as heteroresistance that's been described for Cryptococcus to azoles such as fluconazole. And this is essentially a very small subpopulation of cells, usually less than 1% of the total fungal burden within the patient, that is able to grow on fluconazole. And this is an otherwise sensitive, when you do MICs, population. However, these few colonies actually have a resistant MIC, but will be missed by conventional MIC testing because they are such a small population. So part of Neil's PhD in Tanzania was to do these serial lumbar punctures, a bit like I did in Cape Town on days 1, 3, 5, 7, 14. However, not just plate out onto fungal media, but also plate out onto increasing concentrations of fluconazole, a bit, if you like a solid agar macro dilution, and then count the colonies and then plot both the total and the subpopulation over time, over the 2 weeks. So let's just go back to this particular patient. So this particular patient relapsed after 3 months with headache, fevers, and vomiting, and again was culture positive for Cryptococcus. The MIC was now in the intermediate range of 24. They received the only treatment that was available, again with 1,200 milligrams of fluconazole. After 12 months, there was yet another relapse, and this time the MIC was 64. And this was a patient who was adherent to their fluconazole maintenance. So what did Neil do as part of his study? He sequenced all the isolates from his 20-something patients, including his own. And this is something called a Y-map, which is essentially a chromosomal ploidy map, looking at the number of copies of each of the genes along the 14 cryptococcal chromosomes. And you can see here in this particular patient that on day 0, there's just a single copy of each of these chromosomes. However, by day 90, there is chromosome 1 and chromosome 12 are diploid, and then by 1 year at relapse, there are other aneuploidies. There's also chromosome 4. Now, chromosome 1 in particular is interesting and relevant because it hosts both the ERG11, which is the drug target of azoles, as well as the AFR1, which is an efflux pump. So essentially, what this isolate has done, and you can see here on the right, concurrent with the increasing in MIC, and also the increasing percentage of heteroresistant from baseline, such that by the end, all these colonies, this subpopulation has emerged, are these increasing aneuploidies. And this is a mechanism of genomic plasticity whereby the fungus has this ability, but it's present in a very small population. But under selection pressure of a static drug, this emerges over time and results essentially in this case, gene duplication, leading to treatment failure and ultimately resistance. And what is also important is this is a transient phenomenon because it does come at a great fitness cost to the fungus. So it doesn't maintain them. And if you passage these isolates or you store them in a freezer like we did here in London, over time, then you send them to people as resistant isolates. However, this is lost, this phenotype is lost, and indeed Neil found this. So this is the importance in a way of having access to MICs in real time. And that's where, again, it's fantastic that in South Africa, there is this surveillance at NICD of susceptibility. And indeed, it calls into question this sort of phenotype, if you like, that's missed by conventional MIC testing. What does a fluconazole MIC actually mean? And indeed, this was reflected in the fact that previously in global surveys, including the Artemis survey that South Africa participated in, resistance to fluconazole was said to be very rare. And indeed, nobody was able to show a relationship between baseline fluconazole MIC and treatment failure, although this was largely not in the context of induction with fluconazole. However, Graham had me do a study while I was waiting to set up my initial study at USTA, looking at retrospectively at some patients at USTA relapsing in the context of fluconazole monotherapy. And we found that there was a lot of fluconazole secondary resistance, relapse. And yet when I sent these isolates to other labs because they wanted resistant isolates, they said, well, we find them to be sensitive. So what I suspect was going on here is that there was this transient mechanism that was being switched on and off, this heteroresistance that was selected for. And indeed, looking at Nilesh's data, which he kindly shared with me, it does seem that there is a fluconazole MIC creep in South Africa, despite the fact that since my time in Cape Town, amphotericin B deoxycholate-based induction therapy has been written into the guidelines and indeed become standard of care. And you can see here that over these two periods, 10 years apart, the fluconazole MIC, there has been a creep upwards. And although actual resistance in the sort of range of certainly 64 and above is still rare, there is a worrying shift towards the right. And this is in the context of previously 41% and then 96% of patients being treated up front with an amphotericin B based induction. So that's something to just be mindful of, because we are still using, of course, fluconazole as long-term maintenance in these patients. Now, coming back to Neil's study and in Tanzania, what he was able to show in this series of lumbar punctures is that, coming back to this, looking at fluconazole monotherapy versus patients treated as in Tom Harrison's ACTA trial with flucytosine with fluconazole, you can see here on the left that when you treat patients, and this was based on about 20 patients per in the fluconazole monotherapy arm, you can see here that it does go down like it did in the Nikki Longley study. But this subpopulation, which is the red dashed line, almost meets by day 14 and becomes the entire population, this heteroresistant population, setting the patients up for subsequent relapse with resistance. In contrast, in the 5 patients in Neil's study who received dual therapy with 5-FC, which has a separate target, this subpopulation was suppressed. And indeed, just to briefly show the results of the ACTA trial here, published by Sheila Malloy and colleagues in our group here, where very excitingly, this was a trial looking at multiple African sites and combination therapy using the gold standard amphotericin B plus flucytosine. But this study showed that the blue line here of an oral regimen, fluconazole plus flucytosine, was non-inferior. And this is despite having an inferior, a significantly different rate of clearance, if you look here on the bottom. But nevertheless, this was obviously sufficient to clear the fungus by 2 weeks, or certainly reduce the fungal burden, such that by the time the patients went on to fluconazole, they had a good long-term outcome. And of course, this is very exciting news. And indeed, there are now studies looking at that Nalesh is running in South Africa, looking at combination fluconazole and flucytosine, the so-called EFFECT trial, that will be looking at it for CRAG positive patients as well, using combination therapy. So very exciting to be moving forwards on this. Okay, I come to my second case now, which is a 38-year-old man with diabetes, ischemic heart disease, and renal failure, with a history of transplantation and dialysis, admitted to St. George's Hospital with, as you can see here from the chest x-ray, a community-acquired pneumonia complicated by an empyema, which required transfer to our thoracic surgeons for drainage and eventually a thoracoscopy procedure. But unfortunately, post-operatively, he developed ARDS and was transferred to St. Thomas's for consideration of ECMO. He never had ECMO. However, he remained on ITU for a long period, and we all know such patients. They usually end up with increasing exposure to antibiotics. He also had a pulse of steroids for hypereosinophilia. He developed bacteremias with progressively resistant bacteria, and then he became colonized with Candida auris. You can see here, this was multiple sites. And as Candida auris often is, the isolate was, or more or less always is, it was fluconazole resistant, but sensitive to the other agents. And indeed, at St. Thomas's, this patient received 3 courses of empirica kinocandins for sepsis, query focus, could it be Candida, before being transferred back to St. George's, where he remained heavily colonized despite these courses of echinocandins. Although this was just a single case, we instituted all the known infection prevention and control measures to try to prevent further spread in our ICU, which I'm very pleased to tell you that we managed to do, because I was really worried we would get it embedded in our cardiothoracic ICU. However, the patient then went on to spike a fever, and this rather complicated summary diagram just shows his CRP plot over time and all the antimicrobial and then subsequently antifungal exposure over time. And you can see here that, of course, he had become candidemic, and the line tip grew Candida auris, as did the blood culture. But interestingly, we put him on a nidula fungin, to which we thought the isolate would be sensitive to. However, 4 days in a row, we did serial blood cultures. He remained candidemic. He didn't clear it. So I slightly lost my bottle and doubled his dose of nidula fungin and added in 5-FC, to which the isolate was initially sensitive, but you can see here that, that was initially sensitive. And then he actually cleared it. I switched him to amphotericin B and 5-FC, but by then he actually did clear the infection, and sadly then died a week later, not of the Candida auris, but another, a Stenotrophomonas chest infection. So this is just a typical patient that you probably see more of in South Africa as well, of Candida auris Candidemia, which is the culmination, if you like, of a long ICU stay and all the factors we talked about earlier, and extensive antimicrobial exposure and colonization. So Candida auris has, of course, all the hallmark different mechanisms of resistance that other candidas have, but just more of them. So, of course, we know that drug target mutations in the ERG11 gene responsible for ergosterol synthesis is one of them. You can see here the summary diagram. So the mutations in the target, of course, you can have increased expression of both the targets and then the efflux pumps, CDR1, MDR1. I've talked in crypto about aneuploidy, but Candida too can express either whole or partial chromosome duplications, increasing the copy numbers of both target and pump that again are transient in response to drug. Polyene resistance, of course, has also been described and is less well characterized, but it's thought to be either depletion or altered cell membrane ergosterol, to which amphotericin binds to. And then, of course, there's the echinocandin, the FKS gene, which is the beta-glucan synthase gene. And again, this is a mutation in the target that mediates resistance. So lots of different resistance mechanisms, but also on treatment evolution of resistance. And one does wonder, and we haven't had these isolates from this patient sequenced, although we're going to, but we did look at the serial MICs, and these were not in the resistant range for this patient. However, you can see here on the right, what my postdoc has been doing in the lab is examining a phenomenon that's separate to resistance, again, that's not captured by the MIC, known as antifungal tolerance. And this is a known concept from bacteriology and indeed mycobacteriology, where you get the persister populations. And what this is essentially, again, a transient adaptation, a phenotypic plasticity of the fungus involving stress response pathways such as HSP90 and protein kinase pathways, involving upregulation of all sorts of pathways. But when you measure the MIC at 24 hours, as you can see here by broth microdilution on the right, it doesn't look, it looks like the MIC is 0.25, but if you leave it grow longer for 48 or indeed 72 hours, you see this emergence of this trailing growth or subpopulation of these colonies. And this is known as tolerance. And people have shown that this is very relevant and indeed can be a prelude to developing frank genetic mediated resistance. And you can see here, we all get taught in microbiology about the eagle phenomenon and how you must ignore this trailing growth present inside within the ellipse on an e-test. It's shown here, or within a disk diffusion assay. However, Judy Berman from Tel Aviv has shown in her work that this is actually relevant in Candida, and that tolerance is associated with relapse and more challenging to clear Candida isolates in an animal model. And this is something that, again, we're interested in looking at in humans with Candidemia, whether this tolerance is actually relevant to clearance, poor clearance, or indeed even emergence of resistance. So when we look at Candidemia for England, Wales, and Northern Ireland from the Bristol Mycology Lab, of course, our top 3 species shown in green here are albicans glabrata and paracellosis, and auris is incredibly rare. in the UK in terms of causing bloodstream infection. Colonization outbreaks have been reported from numerous ICUs. However, somehow, even in the context of the recent COVID-19 pandemic, we really didn't have any auris on our ICUs, and I'm not aware of any outbreaks in London ICUs, miraculously, related to auris. However, Nilesh very kindly shared with me this report by Erica van Skalkwijk and colleagues, including Nilesh, looking at Candidemia for South Africa, so of isolates sent both from the private and public sector. And you can see here, obviously, over 6,000 episodes of Candidemia. What's significant here is, of course, that 14% of Candidemias in this survey were Candida auris. And indeed, there was a doubled risk of this if you were a patient in the private sector. Of course, these patients were also older who were in the private sector. They more often, patients with auris, more often had a central venous catheter. These patients, just like the case patient I illustrated to you here, were heavily antibiotic pre-exposed. And also, a lot of these patients had an azole. In fact, with any Candidemia, they'd been azole pre-exposed. Again, coming back to my point about azoles as a driver for resistance. They also looked at the crude fatality ratio, and in fact, there were no differences between the different causes of Candidemia. And indeed, they had this beautiful map in the paper, which really shows where the Candida auris was concentrated, and it does appear, at least for those periods, that it was very much concentrated in the Gauteng region. But of course, there were some reports also from the Western Cape, and both that certain outbreaks, both in public and private sector hospitals, responsible, again illustrating the crucial importance of infection prevention and control, marrying also with good stewardship as a way of controlling this pathogen. And indeed, Nilesh shared with me these excellent guidelines, which I'm hoping you've all already read, which again, we don't have such brilliant guidelines in the UK for management and treatment and a summary of the evidence base about Candida auris. So I would urge you to read this. Of course, auris is not the only MDR candida, and indeed, particularly in centers in the United States, high volume transplant centers that use a lot of azoles and echinocandins, they have seen an emergence of MDR candida glabrata over time because, again, of echinocandin exposure. And it may be David Perlin's work shows from New Jersey shows that indeed, azole pre-exposure may also set up an environment that makes you more liable to develop echinocandin mutations. So again, tolerance may play a role to this. So all these agents, the more we expose these pathogens to them, especially as monotherapy serially, they will evolve mechanisms of resistance. And that brings me to, again, back to combination therapy. I've shown you how in crypto, as many of you who know Tom Harrison know, he's a great champion of using flucytosine. And indeed, through efforts by Nilesh and Angela Loys and various colleagues on the advocacy front, fortunately, we now do have flucytosine available, I understand, in South Africa. And it's also being manufactured by the Drugs for Neglected Diseases Initiative as a slow release formulation. So it may be that we should be thinking of combination therapy, not just for crypto using 5-FC, but also for Candida. And you can see here that this is from a paper by an in vitro study looking at synergy through checkerboard analyses by a group in New York. And you can see here that combinations of both amphotericin B with flucytosine or the various azoles or echinocandins with flucytosine largely did result in synergy with both azole and echinocandin 5-FC combinations being effective even against, in some cases, Candida auris that was resistant to both azoles and echinus. Here, because I have another set of slides on stewardship, just to give you a slight break, because I've galloped through this, and just to ask with Mark, are you happy for me to proceed talking about antifungal stewardship, or do you want to pause here and have a discussion?
Thanks, Tihana. We've got sort of 18 minutes left in the hour. I'm not sure how long your stewardship you were suggesting.
Well, I'll just skim over it. Why don't I do another, can I do another 5 or 8 minutes on stewardship? I'll just gallop through because it's just is that all right?
Please, yeah.
Yeah, okay. So antimicrobial stewardship, I'm sure I gather from Mark, you all did. Of course, we know Mark Mendelsohn is a great worldwide and South African champion of antimicrobial stewardship, but there are certain unique challenges to antifungal stewardship, including the fact that a lot of prescribing goes on in these high-risk patients in secondary care, as we discussed, the expanding host range and the high mortality, which really leads to a lot of overuse, such that it's been shown that antifungal use is very common, especially in ICU patients, and is very often inappropriate, particularly in the case of empiric prescribing. So I'm going to actually skip over this, except to say that at St. George's, where I've been doing stewardship rounds now for over 10 years, again, you can see that hematology and ICU are the big ones prescribing, like we said, but there is a lot of use by other specialties, in particular by surgery and infectious disease. And the other thing, I'm going to skim over this because PKPD is another challenge, but again, is a subject of another talk, but it's much more complex for antifungals. But what is challenging in terms of this prescribing, actually, I'm going to do this slide first, is we know about all this antifungal prescribing ladder, and we really, this empiric prescribing, ideally, we need diagnostics to shift our prophylactic and empiric into preemptive prescribing. But what is challenging for invasive fungal infection, and we are behind bacteriology, is this whole issue of diagnosis. And you will all be aware of the EORTC criteria, recently modified in 2019, classifying IFI, invasive fungal infection, into possible, probable, and proven. And this probable criterion is usually the most common scenario we all deal with, because proven diagnosis is so rare. And here's where we face a real diagnostic gap. We need much better rule in, rule out tests. We've got a great test for crypto, but for other fungal infections, that is not the case. And what we really need, and indeed, we were able to show in our audit of our own antifungal prescribing that about 3/4 of those being given empiric antifungals ultimately have no IFI. And this figure, this was with us not having on-site diagnostics such as beta-D-glucan and galactomannan, but sending it away to Bristol, where it took 2 weeks to come back. So that has potentially not really improved, because we still don't have on-site diagnostics. I'm going to skip over this, except to say that in England, there still is, antifungal stewardship is certainly not embedded. And whilst most hospitals have an antibiotic stewardship, very few have dedicated antifungal stewardship. And one of the key issues in the UK and problems for us that we face at St. George's is access to diagnostics in real time. We send it away to the reference lab. We don't get the result back, usually for a week to 2 weeks in our case, which is very frustrating when you're trying to use diagnostics for stewardships. So we've tried to formulate, for stewardship purposes, these risk scores. You will all be aware of Candida risk scores for prediction in ICU. Their weakness is, of course, they've got decent negative predictive value, but their positive predictive value is, universally poor. But what about if we were to incorporate diagnostics into this? So we have at St. George's developed this Candida risk score, which when you go around on an ICU ward round, people can score this points, and then if they have 3 or 4 points, then they should send off a beta-D-glucan, and these are the patients to consider empiric echinocandin prescriptions in. And indeed, based on the other end of the spectrum of stewardship is, of course, at the stopping end, because as we all know, it's very easy to start empiric prescribing. It's much harder to stop. And that's why at this post-prescription review day at days 5 to 7, we've developed, based on a published Manchester algorithm that showed they were able to reduce consumption and inappropriate prescribing. If the beta-D-glucan and cultures are negative at day 5 to 7, or in the case of suspected invasive pulmonary aspergillosis on ICU, the galactomannan, which now is available also as a lateral flow platform made by IMI, the same people who make the crypto test. Then at that point, if these tests are negative, and these tests have pretty decent negative predictive value, then this can assist stewardship. And indeed, in the UK, we now actually have some, certainly in England, some financial incentives for NHS trusts to audit their antifungal prescribing and to actually get money back if they have done all of these 4 or 5 things. And this has been implemented over the last 2 years. So this is how it's being tackled at a national level to try to improve this in the UK. And there are these rather cumbersome audit forms that we have to fill in for candidiasis and also for invasive mold infection, which, as you'll see here, involves a review at 24, 48 hours and 7 days, which of course in practice we only do it weekly, so we don't do it as often as this, but we try to incorporate these factors. And indeed, again, NLESH has published, embedded within the Candida auris guideline, there is actually a lot about stewardship, including the recommended agents for both prophylaxis and who should have it, and treatment guidelines for the South African context, which of course then need to be locally implemented. There's also a very helpful table looking at the process and outcome measures for stewardship that would be implementable and measurable for the South African context for anyone thinking of implementing antifungal stewardship in particular. And I'm going to skip over this data from St. George's just to move on to my penultimate slide, which is just some ideas for discussion about specifics to Grottescure that I'd be interested in your ideas of. Generic posaconazole, as I mentioned in a couple of weeks ago on this meeting, is now available, and perhaps Sean has looked into whether it's obtainable. Certainly, we are now using generic Anidula fungin here. It's much cheaper than micafungin, and Caspo is also now generic. I mentioned the galactomannan lateral flow device, again, readily implementable on BAL and serum. We're going to implement it here shortly. Bundles, if there's any way that I can help, or we can have some joint stewardship ward rounds via Zoom, very happy to do that. And finally, of course, I wanted to pitch to you this idea where we could together do a clinical trial of combination therapy. And these are just some resources. There's an excellent e-learning from BSAC that I would encourage you to do. It's free of charge. It's online, and also the Antifungal Drug Interactions app from the Fungal Infection Trust. Just download that onto your phone. So just to conclude, that we do need to incorporate antifungals because of emergent resistance. It's it's really important we incorporate it into our antibiotic stewardship. And I hope I've shown you the various ways in which overuse of azoles, but in particular as a class, drives resistance, and that combinations may be a strategy to prevent this. So with that, I'll end. And sorry, it was such a gallop through, and I'll stop sharing my screen, I think.
Fantastic, Tihana. What an amazing talk, and thank you for putting that together and for spending time with us. We do have enough time for some questions. If you can stop sharing your screen, and then if colleagues would like to perhaps put their videos on. I know Tihana quite rightly likes to see people.
Yeah.
And so whoever would like to or is able to, if you could put your videos on. Wonderful to see Nilesh then. Welcome, Nilesh. And I think perhaps maybe the first comment should go to you, Nilesh, if you want. But I mean, while you're thinking of it, perhaps maybe I could just ask Tihana one first question. I mean, we see these cases of cryptococcal meningitis despite antiretroviral therapy and good immune reconstitution coming back again and again at quite prolonged intervals. And we sort of put that, I've often put that down, people have put that down to potentially antigen-specific defects in immunity. How much do you think heteroresistance and sort of drifting MICs to fluconazole might be playing a role in that, as they tend to break through even on fluconazole? Thanks.
Yeah, I mean, do you find when you do the MIC that they are resistant or mostly not? Or do you not tend to do the MIC to azoles in these patients?
Well, again, and perhaps Nilesh can comment, because we tend to send patients up to these samples up. We often don't have paired samples, of course, which is a bit difficult.
I know.
But maybe Nilesh, can I over to you on that, Nilesh? Would that be okay? Nilesh, I can't hear you. I'm not sure if others can.
I can't hear you.
Nilesh, I can't hear you at the moment.
I guess, well, Nilesh, well, yeah, that seemed to be the same problem you had at the beginning, Mark.
Yeah.
Seen that. Yeah.
Maybe while Nilesh tries and figures that out.
Yeah, I mean, the host immune failure, I think that's, you know, Ray Wake, who I know is watching in the audience here from London, who was out working with Nilesh on a cryptococcal antigenemia cohort. As we speak, the PBMCs of patients who are cryptococcal antigen positive and negative from her cohort are being examined to answer this very question, which I think is a valid one, Mark, because as you know, the REMSTART trial from Tanzania showed that patients who have had crypto, despite fluconazole treatment, still have an excess in mortality, even when they're on anti-rets and on fluconazole. So the question is this some sort of genetic or immune host defect, or is this, you know, essentially persistent cryptococcal infection? And I think that is a very valid question. And I think without MICs, you know, how do we know what contribution has resistance? Well, we should do an MIC and look for secondary failure and retreat with AMPHO for those who have it. And of course, in South Africa, fortunately, we are now using, you know, combination therapy up front. So hopefully that's not as much of an issue as the rest of Africa, where monotherapy is the norm. But the only way we can address that, I guess, is careful study. And it'd be very interesting to see in Nilesh's EFFECT trial that is also being run both in Tanzania and in South Africa, whether if you give CRAG positive patients fluconazole and flucytosine, whether, you know, there will be this effect on the differential mortality will be improved. So I think that might answer it. I don't know if Ray, if you are there, but I'm not sure you can actually speak, feel free to comment, or whether Nilesh can come back on.
Great, I think Nilesh, yeah, fantastic. So Nilesh and then Sean, and I think Adrian also had his hand up, but Nilesh, over to you.
Thanks, everyone. Can you hear me now?
Yes.
We can, yeah.
Oh, good. Lovely talk, Tihana. Always a pleasure to listen to you. So I wanted to just say that we've changed the crypto guidelines so that people with a first relapse episode are now recommended to have crypto-MIC tests. And if clinicians suspect resistance, obviously they need to send for MICs. Tihana, I think you mentioned or clearly laid out all the limitations of the susceptibility testing we do using broth dilution. That obviously doesn't pick up heteroresistance unless you do additional tests. But I think a fair proportion of isolates, certainly I'm biased because I see the cases of suspected resistance, but in the reference lab, a fair number of patients have elevated, their isolates have elevated MICs. So resistance is still a concern. And the problem is once you've got someone with crypto resistance, what are the next steps? Tihana, I just wanted to say, I think it's great that you brought up crypto and Candida. I think people often don't think that antifungal stewardship applies to, fungi which are not usually resistant or not commonly resistant. So I'm really pleased that you brought up crypto as well as Candida. I really think that Adrian should make some comments because he was responsible for that excellent antifungal stewardship section in the guideline, in the ORIS guideline. Thanks.
Thank you, Nilesh. Tihana, thank you so much for a fabulous lecture and talk and overview. Can I quickly ask you in relation to the stewardship part in the Candida auris guideline, the conventional teaching regarding the scores, even now for bacterial prediction scores for ESBLs or CREs, was conventionally a rule-in approach, but based on the negative predictive value, there's increasing of opinion increasingly that could or should be used as a rule-out tool to not use the antifungal or to stop the antifungal, or preferably not even start. What is your feeling on that? Is that what you're doing now? Because I went through your list quickly on your slide, but I couldn't read.
Yeah, Adrian, you're absolutely right, because I'm afraid that putting the Candida risk score together, which we did about 4 or 5 years ago, myself and one of the ICU consultants, based on all these scores amalgamated, it's backfired in the sense that my intensivists are sending beta-D-glucans and galactomanins as well on everyone, with sepsis not responding to antibiotics. And there's a lot more prescribing in patients which, as you rightly point out, the PPV is actually poor. And by the time I get my beta-D-glucan back, 2 weeks later, it's too late to have an impact on that days 5 to 7. So what I really tried to do, but have failed so far, is to get beta-D-glucan in-house or, twice a week. Yeah. Because I think you are right. I think people are starting, and where we come in for stewardship is that post-prescription review. So we don't need to tell intensivists to start it. They'll start it anyway. So maybe we should flip it, and we should probably flip it here. And maybe, as you rightly point out in your stewardship, really amazing literature overview, I learned from it as well. That study that used procalcitonin as well. Maybe it made me think, reading your review, maybe we should be doing the reverse and saying, okay, you can start, but if the procalcitonin is, high or whatever, or use the procalcitonin beta-D-glucan if we can get it by then, result together with the Candida risk score at that point, and encourage stopping, because it's that overuse, that we need to tackle. We need to shorten those courses.
But it tends to be in.
No, okay. So Adrian, if you a quick follow up, and then I know Sean's been patiently waiting as well. But please go ahead, Adrian.
No, Mark, I don't think I'm going to add anything, but there is increasingly in the European Intensive Care Society genre also a feeling that these things could be used as a rule out. which I think could be very powerful because the negative predictive value, and you could teach an intensive care nurse how to do a Lyon score, a modified Lyon. It's 4 or 5 parameters. It'll take 2 seconds. Because the problem is the intensivists don't want to use these scores because it's too much for them, you know. So you've got to find some other stewardship tricks to translate that in a rule out.
We
Thanks,
actually
Tihana.
have our microbiology registrars, you know, who could do the ICU rounds, and they often will help calculate it. But anyway, it's like a sort of bristle. Sean,
Thank you.
sorry. Hello.
Hi, Tihana. Nice to see you. Amazing talk. Thanks very much. Really enjoyed it. I have a question for you about these really interesting persister tolerant subpopulations of Candida. I might have missed it. Does that also occur in Candida albicans?
Yes, that's what Judy Berman, my collaborator from Israel, has shown, and I can send you the paper. I was going to put it in, but it was just too much. She has shown that these more tolerant isolates, as measured by various in vitro assays, are more prevalent in those patients who relapse and experience clinical failure in albicans. She's also shown that glabrata, which again I would have loved to have shown you, but it was too much, are heteroresistant to fluconazole. So a bit like in crypto. So there's all these mechanisms that are common to these fungi, and they're tolerant to azoles. They also appear to be tolerant to echinocandids, like I showed you for auris. And this, unfortunately, sets up a path for ultimate evolution of resistance.
Sorry.
We do. And we'd also measure like drug PK, you know, a bit like we did with crypto. So there's whole aspects to this.
Okay. Yeah. So I'm not sure if anybody else, and we're just going over 5 minutes, but we did start late. If there's anybody who'd like to ask a question, one final one from me, Tihana. I mean, of course, you know, the issue about using combination therapy, you know, the potential downside is also to drive resistance. I mean, despite hopefully one protecting the other. I mean, is there any data on that internationally in areas where they've used combination therapy?
Well, yeah, I mean, for fungal infections, I guess you're looking at crypto, where we haven't even looked. I mean, you could say, well, does it matter if you get resistant to 5-FC, if you just use it for the first week or 2 weeks, as long as you're not then going on to that drug? You know, yes, we probably should look at our crypto isolates in patients getting fluconazole and flucytosine to see that whether by the end of 2 weeks there are point mutations in the enzymes. But the fact is we haven't looked, and it is quite a short course. And the same would apply to Candida. Whether that could drive resistance, I don't see how combining with 5-FC could drive resistance to echinocandins, you know, which are fairly cidal. But I guess the theory being that it might protect them from evolution of resistance. So I'm not aware, and again, in Aspergillus, no one's really looked, and we are interested in that, you know, combinations for Aspergillus as well. But the answer is no one's looked. Everyone's focused on azoles in the setting of monotherapy.
Look, it may protect, but you know, in the end, the more of each drug you use, the more potential there is to drive resistance, and maybe not in the target fungus that you're trying to treat, but in, you know, if you're going for crypto and you're affecting Candida or you know, or other fungi. So no, that is a very interesting sort of broader, zoomed out perspective. And so lastly, and sorry, I did say lastly, but I'm going to ask one last cheeky question. Being a great proponent and advocacy of access to antifungals globally, perhaps your 1 or 2 sentence take on what's happening in India with mucormycosis.
Oh gosh, yeah. I mean, it's interesting because we had a science media query the other day, but I was too late for it, asking about the black fungus in India, and did anyone want to comment? My take is, I mean, it's absolutely tragic what's going on in India, but particularly you take a population of lots of diabetics being treated in the community in a tropical climate. You know, I showed that slide, exposed to all these fungal, and you chuck in some steroids, you know, moderate doses of dexamethasone, and some poor glycemic control, which they induce, and you get this in. And we know that there is a lot of mucor in diabetics in India anyway, and aspergillus. They seem to get a lot of it, even in apparently immunocompetent. So unfortunately, it's led to an explosion. It's a sort of perfect storm, isn't it?
Yeah.
Steroids, diabetes, India.
But
then the drug runs out, the drug amphotericin.
Yeah, well, exactly. That's the issue with that. I mean, saying that, you know, South Africa also has a heck of a high poor diabetic control population getting high-dose steroids with COVID. And I mean, maybe we're not tropical enough here in Cape Town, or, you know, the conditions aren't right for mucor, but the loss of amphotericin.
Another reason we need posaconazole. We need to get those tablets, so we've got an alternative for when, you know, when we need to treat alternative agents.
Tihana, sorry to throw that little curveball in at the end, but I know you're very passionate about access to antifungals, and, you know, the amphotericin story now coming out of India is
I know, especially as you know, the Indians make their own amphotericin.
Exactly, yeah.
I think it's called Amphosil. They make so much stuff. It's just a tragedy, really.
Tihana, thank you, you know, from all of us for spending time with us. Thank you to all your registrars for joining. I'm sorry we can't see them behind the door or the lecture theatre they're in. And as you say, maybe there are options for joint meetings. And it's been wonderful to hear, and fantastic learning for all of us. So really appreciate